Hello. Eukaryotic transcription is a complex process, and there are many things RNA polymerase has to mitigate to make all the transcription and also the processing of mRNA at the same time. This is complex, and there are multitude of proteins and factors involved in doing so. But there is a sequence of order that things happen, and that's what we will try to see now. Assume that this is a gene that is given, and that's assumed that it's a part of a chromatin. We are not discussing about nucleosomes at this point. So if this is the mRNA, if this is the DNA, and here we have exon 1, exon 2, exon 3, and exon 4, we have intron 1, intron 2, intron 3, and we also have a polyase signal sequence, and here we have promoter. So when RNA polymerase starts out here, and then it encounters the RNA is starting to emerge, capping should have occurred, or capping enzyme should be recruited, and then the intron has come, it has experienced an intron. Then we need genes, or the proteins that are responsible for cutting out the introns and splicing the exons. So here we have the 5' side of an intron, and in this case we have, at this point we have 3' side of the intron. So as the RNA polymerase keeps moving on, it is experiencing, or it is encountering, or introns and exons coming, and at the same time the processing of RNA should occur. The RNA that is being produced at the end should have a G-cap, should have all the introns removed, and at the end, and the 3' side, it should have a poly-A tail, and then transcription should terminate. These are the sequence of events that should happen. It starts with promoter escape. From then on until the termination of transcription, there are a sequence of things that happen. And how does RNA polymerase make these things happen? It has to do with the tail of RNA polymerase, as we have seen before too. From promoter escape, even many other aspects, the CTD, carboxylic terminal domain of RNA polymerase, is important, and especially within them we have several sequences, a motif like YSPTSPS, which is repeated multiple times on the domain, and the phosphorylation state of this motif will determine what proteins will bind onto the tail, and then from the tail they will hop onto the newly emerging mRNA. So it's like all these proteins will keep coming onto the tail, and from tail to the RNA, based on the phosphorylation status of this motif. So say for example, if we have this is 1, 2, 3, 4, 5. If the 5th serine is phosphorylated, it will promote, or at the stage of promoter escape, the capping enzymes are recruited onto the tail, and as soon as the mRNA emerges out of the RNA polymerase, the capping enzymes will hop onto the mRNA, and that is how capping enzymes are first recruited. And at a later stage, if during the elongation, where we have seen that the second serine at position 2 is important, phosphorylation of it will facilitate elongation, and then at the same time it will recruit these proteins for splicing. Say for example, if we have at this stage of in this stage, we can assume that we have the promoter escape, and then the capping enzymes would hop onto it based on the 5' serine phosphorylation. As soon as the mRNA is coming out, the enzyme will hop onto it and will start capping the 5'. And when the RNA polymerase has moved on to the elongation state, now the serine at the second position would be phosphorylated, which will recruit the splicing machinery. First, it will recruit the 5' splice proteins that recognize the 5' splice site, and they will, as soon as the mRNA, this intron for example comes, those proteins will jump onto the mRNA, and then 3' splice site recognizing proteins, etc. And this is how the transcription will continue until it reaches a poly A signal. Poly A signal sequence, when it is transcribed, is bound by two proteins called as CSTF and CPSF. CSTF and CPSF will initiate the termination of transcription, and then there is further processing. The mRNA will be cut, some of the parts will be left out, and that will allow the binding of another protein called as PAP, or poly A polymerase, which will add hundreds, about 200 adenine molecules to the 3' end of the mRNA. And this poly A tail is also bound by several proteins called as poly A binding proteins. So the 5' end of the mRNA is protected by the G cap, and the 3' side of the mRNA is protected by the poly A tail. And there are many other functions that something like mRNA could bind upon itself, something like this, the 5' end being here and 3' end being here. So the proteins involved in translation, initiation, and the poly A binding proteins can interact, so the mRNA is in a kind of circle which will keep on rotating or recycling the ribosomes after initiation, elongation, termination, and reinitiation is made possible. That way, from one mRNA, there is efficient production of proteins. So the differences between the prokaryotic mRNA and eukaryotic mRNA, let's review that quickly. So GCAP, all the majority of the post-transcriptional modifications are in eukaryotic mRNA. The GCAP, poly A tail, and the splicing of the exons. We should not say splicing of introns. Splicing means joining, so we should talk about splicing as joining of exons rather than cutting away of introns. Cutting away of introns doesn't imply joining of exons, but if you say joining of exons, which would imply that elimination of introns. So these are the three important differences: capping, intron removal, and the poly A tail. And another one, the important one, is that prokaryotic mRNAs are polycistronic, whereas eukaryotic mRNAs are monocistronic. We'll see you next time. Thank you very much.